Sure. Just let me share my screen here. Okay. So, good afternoon, everyone. Amber and I are going to talk today just a little bit about how we can get science moving from the lab to clinical trials and get
Interested or not, if not, let me know. We'll take it over.
and get those medicines, the
we'll
latest, greatest novel medicines to the patients that need them. As researchers, we often get posed very important questions regarding PD research, like why haven't we solved Parkinson's yet? And how do we study them in the lab to even understand Parkinson's? But of course, the most important question is how soon will these new treatments be available? Often heard as why do we even need clinical trials? So what we'd like to do today is try to impress upon everyone what kind of things we do in the lab to try to understand PD and the importance of these clinical trials in getting these new treatments out. There we go. So to answer the first question, why haven't we figured out Parkinson's disease yet? And the simple answer is it's complicated. For many years in the lab and even in the clinic, Parkinson's disease was primarily viewed and researched as a movement disorder, looking at the outward symptoms that the patients express, the bradykinesia, the resting tremor, the posture and gait. But in recent years, and I mean recent years, we've really understood that it's a lot more complicated than that. There are psychiatric issues that can come along with Parkinson's disease, such as sleep difficulties and depression and dementia, and increased risk-taking behavior. There are also autonomic disorders, so symptoms that really do impact daily quality of life, but aren't outwardly obvious and may not even be heavily talked about when it comes to the clinic, and definitely not looked at when it comes to the lab, such as gastrointestinal problems and heart problems and blood pressure issues. So when it comes to trying to tackle Parkinson's disease and to get better treatments, both researched in the lab and then out to patients, we are trying to now change how we look at the disease so that we can tackle more, if not all, of these symptoms and truly treat the whole disease and the whole person, or get better treatments that focus on these individuals.
I was going to just say, I mean, it's a whole body disease. It's not just those few little cells in the substantia nigra that are a problem. It's the whole body is affected, and it's really changed the way that we've conducted studies, in the lab and all the way back through now.
So in order to try to find new treatments for the disease, we really need to understand what causes Parkinson's disease. But what does cause Parkinson's disease? And unfortunately, the question remains, we don't have a good idea of how Parkinson's disease is initiated. We know that it's a progressive neuronal degeneration. We know that it can affect the whole body and the whole brain. And we are learning more every day as we develop better models in the lab, as we conduct more clinical trials, as we study more patients. We know what kind of pathology to look for in the brain. We know what kind of symptoms to look for in the patient. But these are all telltale signs, and we're still working to get back to that ground zero where things start so that we can attack the disease there. Now, when it comes to how Parkinson's disease starts, one of the struggles that we have as researchers is the fact that most cases of Parkinson's disease are completely idiopathic. In other words, we don't have any idea as a researcher or a doctor where the disease started. There's no link, no genetic link, no toxicity link, no environmental link. Many patients just suddenly present with Parkinson's disease. And there doesn't appear to be any link as far as race or fame, as you can see in the top corner here. Some populations, like farmers, have a higher risk that's associated with chemical exposure or industrial workers. And then of course there are the familial cases or the genetic cases. So there is increased risk if there's a family history of Parkinson's disease. And we don't have a good handle on what that risk is for every single gene out there. But for the truly what we call monogenetic cases, the cases where this gene is associated with getting PD and it gets passed on, and this one gene can increase your risk for PD, that still only accounts for about 20% of all Parkinson's cases. And then as I mentioned just a little bit ago, we have the environment and toxin exposures that have been linked with an increased risk of PD. That doesn't necessarily mean if you're exposed to these things, you're going to get PD. Like when I mentioned farmers earlier, farmers that work with very specific herbicides or pesticides, such as rotenone or paraquat, have a 4% increased risk of PD over the idiopathic population. So it's not a huge increased risk, but it is an increased risk. So where we're left at in the lab trying to study the disease, our current ideology suggests that most PD is likely starting due to a combination of genetics and environmental exposure over lifetime. Some people may just be more predisposed to getting PD. Some people may be less predisposed to getting PD. And depending on what things, where their life has taken them, what things they've been exposed to, is going to tip the scale on that risk of getting the disease. But again, we're talking about risk here. We still don't have a definitive, if this and this happens, you get PD. We don't have that. And we're still trying to figure that out.
There's actually some of the genes actually within even twins, like it can look completely different. Like someone can have the same exact mutation. One goes on to have Parkinson's. They've been therefore in the same environment being raised, and yet the other sibling would not have anything or mild symptoms. So it's just, again, it just adds to what you were saying earlier with the complicatedness of PD.
There is one case in particular. So many of you might by now have probably heard of the LRRK2 gene, which is very highly linked to developing PD. And there is a case of twins where both of them have the mutant gene. Both of them are in their 60s. One has severe PD and the other brother has nothing. And there's been several papers written about them that's being studied. So again, we're talking about risk, and we're trying to figure out in the lab how those risks add up and where we can start with a treatment that'll mitigate those risks. So how do we develop and study models of PD and treatments? Well, the first step is getting a model. Right? We can't constantly bring humans in and dissect them and look at things. We need a way to model Parkinson's disease in the lab so we can test every possible idea that we have and get better ideas of what's causing the disease. Once we understand better what causes the disease, how the disease progresses, then we can start identifying new therapies to try out to slow down, stop, or even reverse the disease. But what this boils down to is being able to find the right model. So an ideal model of Parkinson's disease should meet several criteria. It should exhibit the disease hallmarks. So the pathology, the alpha-synuclein accumulations, the neuron loss, the movement disorders that are associated with the gastrointestinal disorders. Those need to be present in a good model. It should be predictive. We know how the disease progresses in humans. We should have a model that progresses the same way, where we have progressive neuron loss, where we have progressive movement dysfunction. That way we can actually develop treatments that are neuroprotective and can stop this progression. And the fact of the matter is, we're still trying to work on the ideal model. We don't have one yet. We have ones that are really, really close, but they still involve things like very altered genetics or direct exposures to toxins that a human normally wouldn't be exposed to. And while those get us close, while those give us ideas, some of which have translated to clinical trials and even treatments, not a lot.
But this goes back to the first slide. I mean, a lot of the problems here is that these models have been focused on the motor aspects of Parkinson's. And so we've gotten pretty good at treating the motor symptoms of Parkinson's and not the everything else, which can actually be more of a problem for a lot of people. And, you know, it's when you can't even start with a good model, it makes, you know, all of the research that comes thereafter it impacts the way that it turns out. And if you aren't asking the right questions, you're not going to get a different answer. And I think is part of the issue in making headway in better treatments for PD.
And another very important part of this having the good model is this stuff needs to be translatable to humans. So, and this isn't just for Parkinson's disease. This is for every disease that gets studied in research, cancer, Alzheimer's, everything. You know, there's jokes within the lab that we've cured cancer and Parkinson's in mice 10 times over, but you know, the cures for mice don't always work for humans. So we need to keep working on getting better models so we can get better treatments that actually will do what we want them to do when we move into the clinic. So what are some of the ways that we've been modeling PD in the lab? Well, the most basic that gets us our first ideas is just cell culture. And this is literally what it is. It sounds like it's growing cells in the lab that look like or model the neurons in people. Here in the corner, I have an example of what are called Sy5y cells. These are an immortalized cell line that can be turned into a neuron-like cell. And generally, cell culture is used for like high throughput screening. So you do a genetic change or a toxin exposure to the cells on a dish, and then you can throw hundreds of different drugs at them and study what each drug does really quickly to try to get an idea of which ones might help mitigate things like oxidative stress or cell death. But that's a first step. We would never go from cells to people. The next step is to actually make sure that this works in an intact organism. So then we move on to our animal models. The most traditional models that most people are probably familiar with are rodents and monkeys, rats, mice, non-human primates. And one thing that's very important to stress here is that usually a study involves multiple different kinds of animals to make sure that a treatment can cross between different species. If something only works in a mouse but doesn't work in a rat or monkey, it's probably not going to work in a human. So we create models that simulate Parkinson's disease. You know, rats and mice tend to be used for genetics or toxicity models. Monkeys are usually used to determine the safety or for surgical purposes to determine drug delivery. And we do have models for non-human primates as well that we can use to simulate Parkinson's disease. But what about that high throughput? If we do a super high throughput in cells, we obviously can't test, and we get like 30 drugs back out of the cells. We obviously can't do 30 different kinds of studies on the mice. That would be way too many animals. So there are now non-mammal models that allow us to do this high throughput in an intact organism. So things like fruit flies, zebrafish, which I worked on when I was at Pitt, or small worms or nematodes. This is a way to look at moderate to high throughput screening of drugs in an intact organism, maybe to whittle things down. Zebrafish have the added benefit of being a vertebrate, like mammals and humans. So evolutionarily, they're a little closer to us than, say, a fly or a worm. In all of these cases, cells and the animal models, we usually try to generate a Parkinson's-like state or Parkinson's-related dysfunction, and then we try to treat that dysfunction. And then the next step after that would be trying to get this into patients. So the typical ways that we simulate Parkinson's disease, there are the genetic models. As I mentioned, there are some genes that have been associated with PD. Even though they don't account for a lot of the cases, The pathology for most of the genes is very similar to idiopathic Parkinson's disease. So it gives us a clue of things to chase after for therapies. So cells or animals can be genetically modified to have these same genes or lack these genes that Parkinson's patients have. And then we can study to see if we can treat that. And then there are the neurotoxicity models, things like the pesticide rotenone or the herbicide paraquat or the industrial chemical TCE that cells or animals can be exposed to simulate Parkinson's disease. And then we can try to treat that and see if those drugs can translate. And so then there's some of the things that we do when we have these models to generate these new lists of therapeutics to start studying. I mentioned the small molecule screening. So typically this is done in cells or the smaller non-mammalian models like fish that allow us to screen a whole library of genetic drug, gene therapies or drugs or small molecules rapidly and get some ideas. And then rats and mice and the primates tend to be used for the investigations once we've whittled this down to one or two ideas and we want to look at the best way to deliver it, the best time to deliver it, how does it really affect disease course, things like that. But at the end of the day, the ultimate goal of all of this work is trying to jump from what's in the lab to the patient. And as Amber mentioned early on, for decades, up until very recently, most of the research was focused on the movement disorders. So a lot of the current available treatments don't address the progression of the disease. They address the symptoms, primarily the movement symptoms. So a lot of the research that I'm talking about, a lot of these models are generating, a lot of these small molecule things, we're turning our attention to try to modify the disease, to slow it down, to stop it, and to do that over the entire body or the entire brain, whatever the case may be, depending on the therapy. So really, how do we jump this canyon? And I'm going to turn it over to Amber to talk about the actual clinical trial side of things.
So this is usually called the valley of death, meaning that you have cured Parkinson's many times in mice and rats. And yet, once we try to make this leap, across and into the actual patient itself, pretty much, a vast majority of these things have not made it. They've failed to make that leap. And so it makes it, really hard to not get so excited when you see the news. And Dave was just texting me about another late breaking thing that sounds amazing. But again, like once you actually start looking at this in a really rigorous way by doing formal clinical trials, we unfortunately, especially with Parkinson's, just haven't been all that successful in making much headway in really getting at the basics and the problems of how this disease is even being generated. Can you go next slide? So I just wanted to cover just some of the basic mechanics of how we do clinical trials. I'm hoping people have heard some of this, and I've actually found, talking to my own dad, with COVID and watching the rapid development of the vaccines, that I was actually really pleased in that it was really helping to educate a really broad population as to what are clinical trials and how you step through that process. But what we were just talking about is all this discovery part. So we often start at the level of cells in the dish, and that would be here what they have here is the studies in the lab. Next. Now, fortunately, if there's something that looks really good from just an understanding of how the drug may work, like what it actually does inside the cell, and you want to step it up and put it into an animal living organism to see how it's affected, and something with a beating heart and eats things and plays with other friends, right? All these things impact, like especially neurodevelopment. What happens when you then apply your drug into a living animal? And I think most of us here probably would say that they would rather do this in animal models like rodents first before jumping right from a cells in a dish into people. And this is traditionally how we've been doing this for many, many years. Next one. So sometimes there's a phase 0 study. So in some people, they will actually do this in healthy controls where they will give a particular type of drug just to see dose, make sure it's safe, see how the body's using it up. Can they detect it in the blood? Can they, are they getting levels that, that they were expecting? Just really basic information like this. And oftentimes, especially when it comes to Parkinson's research, this can be done in healthy controls. So there's a few studies that are out there now looking at something like this, just to understand the drug better in people. And then we have our phase 1. So this is where a lot of Parkinson's studies have been started. Unfortunately, it's really hard to go from phase 1 to phase 2. A lot of things fail here after phase 1. But generally, your phase 1 study it's a smaller number of patients, depending on the type of thing being investigated. It can be very small. So I know for like gene therapy studies, for example, we're talking about like a dozen patients being involved, whereas like some other drugs may be more like up to like 50 or so. Good. The purpose of the phase 1 is really is oftentimes finding the right dose for that patient population you want to be going into. So if you want to be looking in Parkinson's disease, this is when you'd be first introducing that new treatment into a Parkinson's patient. Again, it's really focused on safety. So the goal for phase 1 is to, how safe is it? How tolerable is it? Are there a lot of side effects that pop up or not? Or is it going pretty well? And is it looking like it's okay in Parkinson's, like specifically, whereas in phase 0, that could be in healthy controls. So again, in phase 1, we're looking for potential benefit, but the main goal for phase 1 is really purely, is it safe? And if we feel like the FDA says, okay, you get the go ahead, it looks like this is safe, then you can go on to do a phase 2. So phase 2, unfortunately for a lot of Parkinson's drug, this kills off the other large chunk of trials that have come through and made it this far. But this is where you really start to look at whether or not your drug is actually having any benefit. So it really becomes important as to what you think your drug should be doing and how it should be affecting Parkinson's. And again, in a lot of cases, a lot of our medications and treatments that are under investigation are still really focused on is it improving motor scores? But there are a lot of things out there now that are still looking at constipation, blood pressure control, these other things too. And it's really at this stage where we want to see if it's actually effective in doing what you want it to do in your disease population. And because you're trying to ask a question of effectiveness, this is why, just from a statistical standpoint, we need more and more patients in order to be able to satisfactorily answer that question. Again, really coming down to the statistics of it. This is where it's, I think, becoming a lot clearer that we need to be better at designing these studies specifically for Parkinson's. In that, I don't need to tell anyone here, no two Parkinson's patients are the same. Depending on your age and whether, other diseases you might be having or other medications that you're on, or if there's family history or other exposures to toxins. I mean, these are all factors that are going to potentially change, how Parkinson's looks for you and how you may respond to something. And so I think there's, I can see definitely an interest in really what we could call homogenizing the population. So really trying to pick like a section of Parkinson's to really focus in on, a subpopulation of Parkinson's patients that we think may be more beneficial for a particular type of medication. This is a really key step in making drugs available. And then if things pass with phase 2 and it's looking good there, then it goes on to phase 3. Now, phase 3, this is really what you're seeing here is really the traditional stepwise fashion for how we move drugs through clinical trials. And traditionally speaking, phase 3 is you are now taking this medication or some other drug, other treatment, and now you have to apply it to a very broad population. So you cannot look at this narrow segment, you're looking more broadly. And does your effect that you thought you saw, does it hold up now when you're looking at a much larger population? And this is really where we get a good sense of the side effects, because everything has a side effect. One of my favorite questions, well, does that have a side effect? Like, yes, of course it does. They all do. Every single drug has a side effect. But what kinds of side effects? Because, having, I don't know, feeling cold from your medication versus losing a toe. I mean, these are completely different types of things. And this is really what we're trying to do with the phase 3 is what are those side effects? How bad are they? How many people get them? How many people don't? And this is really the big hurdle. Once you get past the phase 3, this is when you go to the FDA and you say, okay, look, we got all this great data. This is what it's doing for us. We are now asking for this to be an approved medication. And even if you get past that, there's actually a phase 4 where once the drug is technically approved, it is out in the world, it's being prescribed. There is actually still further follow-up after that to now that you've got this out in a much larger population out in the world, how is it behaving? What are the side effect profiles looking like they did when you were doing the actual phases earlier on? So there's still more due diligence that needs to happen even after the approval may be given by the FDA. And I think, again, what you're seeing here is a very traditional way of doing the stepwise fashion of clinical trials. And there's really actually, I'd say, an urgency and desire too by the FDA to skip this, or not skip it, but to modify it so that we are trying to be more be a little bit faster with this in a safe way still. But sometimes you can combine your phase 2 and phase 3, or you combine a phase 1, phase 2. So there are ways of designing these studies to be a little more efficient. And this is really a big trend that we're seeing now, and arguably, more important for things like Parkinson's, where, patients are just getting worse over time. And to really try to get to these treatments that are really going to impact the disease for PD patients. Go ahead, next slide. So it's interesting having lived on both sides of clinical trials. What are some big takeaways that I would want to share with anyone who is maybe thinking about doing studies or being part of a study? I think the real key thing, and I'm really going to really hammer this home today, is that none of this is happening without people like you volunteering and trying to participate in these studies. Nothing goes if we don't have that. I think the other thing to really remember is that safety is always a priority. So, even if we're talking about the phase 3, still safety is still number one. We do everything we can to really make sure that these are done in a very safe way. That being said, it's still an experiment. And so there are still risks and benefits to being part of any study. And this is really the key thing that I would encourage anyone that's considering to go into a study. Those are the things you really need to focus on when you're talking to the investigators, your doctors, these sorts of things. What are the risks and benefits of anything you might be interested in looking at? And to keep in mind that there are a lot of clinical trials that actually don't use a drug at all. And in fact, this is the way it is for a lot of diseases that have no cure and no treatment. A lot of them are what we call observational studies. So simply, what happens in a disease as it progresses over time, with people just living their daily lives. And there's still a lot of value in those sorts of things. I know, like the Michael J. Fox Foundation, especially with their, they were doing this PPMI program, which I'm sure some of you may be aware of or participated in, but it's also looking at biomarkers. So, is there something that we can learn from, your genetics, and then also the genetics of, everyone else participating, that will help us generate new drugs and new treatments in the lab that we can then step forward through this process. So to keep in mind, there's a lot of value to studies that don't use any drugs at all. That being said, for some people, there are a lot of studies that have a placebo, and this may not be appealing to some people. But again, there's really no better way for us to understand what's Parkinson's and what may be, just like progression of the Parkinson's disease versus a placebo effect. So it turns out that the placebo effect is really, really strong in Parkinson's disease, and really just because it's dopamine that's getting messed up, and it's dopamine that's involved with reward. And this is why we think that there's a really strong placebo effect in Parkinson's patients involved in these studies. So just to keep that in mind as a potential issue that you need to be considered when you're talking and thinking about a clinical trial. I've said this before, and I will keep saying it, no legitimate trial will ever ask you to pay for anything. You should really be concerned if you're asked to pay for anything upfront. These studies should be well funded enough to be able to do everything that they say that they want to do, and you should never have to pay yourself. And again, you can confirm this whether or not they are registered on clinicaltrials.gov. So I have the website listed here. And I would encourage people just to go look at clinicaltrials.gov, see if there's anything that may look good. I have some other slides a little bit later on with some other resources. Okay, go ahead. Okay. So, like I said, even if you aren't interested in getting poked and taking strange drugs, again, there really is a fit for just about everybody, and this goes beyond Parkinson's disease. Even spouses and family friends that could even participate as controls for some of the Parkinson studies. And again, there's just observational studies out there. There's some that are really short. There's some that are really long. I mean, there's something out there for absolutely everybody. And sometimes it's just a matter of finding the right fit, and just even think about it. And if it's something that you think you might be interested in and that you might want to participate, this is where you need to talk to your doctors. Sometimes they won't bring this up in the clinic if you're not bringing it up yourself. And so if you start the conversation, a lot of times that'll be a great way to get started. Let them know that you're interested in thinking about something like that. And maybe they have an opinion on something that's going on. They, or may know of someone else for you to talk to, but also let your family and friends know that you're thinking about participating. And if you do find something that looks good, I always encourage patients to talk to their families and friends about the pros and cons, because these are things that it has a potential to impact the whole family. It's not just the patient with Parkinson's alone. This is really a family affair, in my opinion. So, and then just some general things to kind of keep in mind if you're thinking about participating in a study. So, are there other medical conditions that you have right now, or other medications that you may be on that may interfere with whatever it is that the other study may be looking into? Or even if it impacts your life, and with whatever medical conditions may be there, maybe a study is not the right thing, or maybe it changes what kind of study you go into. For some of the studies that we're doing, we're looking at a really, like, as early as possible disease state. And so for a lot of these folks, it's, they're younger and they may be working, and sometimes being very busy, running high level jobs, that sort of thing. Sometimes that doesn't work really well with some types of clinical studies. And sometimes you get these, jobs that really want to help you make sure that you can participate in these studies. And so I think it's important to kind of keep that in mind when thinking about participating. And then it's just the time overall. There are some studies that are very intensive and require a lot of visits, and there are some that really are more minor. You can do them online, or it's just a video chat, things like this. So just to keep that in mind, that not all studies are the same with that. And again, I think really one of the key things here is not everyone has the same level of risk that they're willing to take on. And, to keep that in mind when you're looking at studies, and kind of, again, going into this idea of how would you feel if you got into the placebo group? Is that going to be a problem for you? Or do you think that this is an okay, risk to take in order to help the science move forward, essentially? And again, as I said before, just think about how this or doesn't impact your family, friends, and just the overall lifestyle. But just a few suggestions of things to think about if you're considering joining a study. Next one. So, man, the first time I saw the statistic, my heart sank. I was like, "Oh my gosh, it's true." So, I feel like I would have said the same thing. Yeah, a vast majority of patients with Parkinson's would say, "Yeah, I would join a study for sure," and yet in reality, go ahead. It's actually less than 10% of patients actually sign up for clinical studies, and I think it's a harsh reality in a way when you hear from a lot of patients that they really do feel the importance, they understand the need for doing this kind of research, and yet I think it's important to know that it's really the vast minority that actually will sign up and do this volunteer work. Next one. And so why do we want to do this? The environment that we live in right now, I think there's some folks that would be interested in like just jump straight into people, like let's just do this work, let's just make them all approved and we'll figure it out later. But there's some benefits here to doing really rigorous studies, make sure that we know we've got safe drugs going out on the market. It's, again, I can't say it enough, but cannot replace the actual volunteers joining the study. There's no amount of money you can throw at a study. If there's no volunteers that go into it, it doesn't go anywhere and we learn nothing. So again, really the importance of keeping, the idea of potentially participating in a study as reality. I hope to encourage a few people today to think about it. And so this was another statistic from the Fox Foundation that 85% of Parkinson's, specifically Parkinson's clinical trials will be delayed, with 30% of them actually never making it into completion for even a phase 1 because they just can't get the Parkinson's patients to volunteer. And it's really without the altruism of volunteers, we just will never find a better treatment. Next one. And so I like this graphic too. I mean, without trials, we're never going to get new drugs on the market. We're not going to find, that the next big drug that's really going to impact patients. And so and that being said, a lot of patients that I've worked with, they're not doing it necessarily for themselves. They know that they're doing it for everyone else that's coming behind them and everyone else that maybe get diagnosed with Parkinson's the next year. And this is how we make a difference for them. But another key thing is, the more robust we make these clinical trials, it helps us go faster. It helps get to that point where we get something on the shelves a lot faster. And this is how we find new things and make breakthroughs and make a real difference in what we're doing, and not just a new iteration on Levodopa. This is how we make new therapies really come to life. This one. So please think about joining the fight. Again, that the cure we all want to find, it's never going to happen if we don't have people participating in the research. So just think about it. Talk to your doctors, talk to your family. Go to the next slide. So I already told you about the clinicaltrials.gov. I think I mentioned almost every time that we do one of these talks. Just browse on there, just type in, you're looking for Parkinson's studies. I mean, you'll get to see even the history of studies that have been out there. A lot of them are still on there to be searched. You can see what's there. They also, in some cases, will even show you some of the data that came out from the studies that were there, and if it worked or it didn't. And it can also give you a sense of like what phase are they at? Are they at phase 1? Are they at phase 2? And that gives you a sense of like how far along they are, how advanced the studies are, if there's placebo, if it's something that you maybe shy away from, or something that's actually really exciting. And again, every study at this here in the United States must be registered on clinicaltrials.gov. Occasionally you'll see trials that are registered here that are only done in Australia or Europe. They sometimes will show up here, but this is really one of the best places to look. And the next one. honestly, if you want to look at just Parkinson's specific studies, Fox Trial Finder has done a fabulous job to put together all the studies. So in fact, like having worked with the Trial Finder for our studies, they do a really good job. They actually pull everything off of clinicaltrials.gov that's Parkinson's related and put it right here. And they actually can you can actually register and just give them some of the basics of your Parkinson's disease, and they can actually send you ideas of studies that may fit for what is it you're looking for, what you're interested in. So if you haven't signed up and just registered, I'd really encourage everyone to do that. Because again, there are a lot of studies that, especially in the middle of COVID, that were started that are just simply, just FaceTime on a screen, or maybe like wearing a Fitbit or something like this, so that are really minor, but yet still is giving back to the community, still participating in research, and still helping us get some answers to questions that we're all still asking ourselves. So I think that's it for our slides. So I was hoping we could go into some questions now. We just saw a question pop up. Let me see here. Let's see. So there's a question for David about whether or not we could get information about trials to be passed along. And I gotta say that the Fox Trial Finder does a really nice job of doing that for you. And it can actually customize what studies are being sent to you if you can like fill out the profile. There's also, I'm sure many of you have been on the Fox website. They have a really nice, just a booklet on like how to get involved in clinical trials and things to consider, and other ways for advocacy. They've done again, they're research based, and so these things are very important. So they've done a really nice job of putting together information there. There's also some nice little videos to consider as well. We can happy to send that website around too.
Excuse me, I just regarding that question, in Casey's June newsletter, there's a recap on some of the studies that are ongoing at Pitt with contact information there.
Yep. Again, just given all of the remote capabilities that we're building into some of the studies, think about looking at things that are not happening just in Pittsburgh, because anymore this is doable in a more broad way, which is really exciting. And I think that was one of the benefits of going through COVID is how it's really changed what we do with these clinical trials and making it available to people who are maybe not living in the same city as the investigators. Thank you, Casey, for putting that up there.
You're welcome.
Anything else you wanted to touch on?
There was a question about Parkinson's disease and traumatic head injury. It was the first question that came in.
I can talk about that a little more broadly. So, I mean, it's the link between like head injury or trauma and Parkinson's is not so straightforward. I think it's really easy to say that if you've gotten hit in the head enough, it's not going to help reduce your risk of Parkinson's. I think everyone would always, automatically thinks of Muhammad Ali with being hit so much. So yeah, I think that there's an increased risk with a lot of head injury specifically. But then again, like a lot of people get hit in the head and never develop Parkinson's. So it's not such a clear, direct link. But if you could avoid getting hit in the head, I would really encourage that. But that being said, it's not just Parkinson's that has had this link. There's other neurodegenerative disorders that have had similar type of links to head injuries as well. So I think it's a topic that we're curious about, but it's not so clean. It's not such an easy answer to have. It's certainly not part of the models that we use in the lab. I'll tell you that much. There's yeah. Okay, yeah. So there's a question here about Nuplazid. I can read it out loud. So my husband was on Nuplazid for a hot minute. We had to discontinue after a week because it made him super tired and lethargic. Do you have any info on the effectiveness and safety of Nuplazid? So this is, I really like Nuplazid in some people. Like it can actually do a fairly decent job. But you know, as we've learned from doing these studies, you know, not everyone can tolerate it. It's also really hard when the people who would probably need something like Nuplazid are already, you know, it's a complicated disease state just by even needing that. And so, you know, I think it makes it hard to know what's the disease and what's the side effect from the drug. But, you know, clearly it was considered to be effective enough that it became an approved medication. And, you know, there's a black box warning around Nuplazid because of the risk of death that's associated with it. But a lot of the medications that we would use, like Nuplazid, so sometimes this is for people who are having hallucinations or seeing things. Again, this is usually only occurring at really late stages of disease when people were already not doing well from other respects. And so that's one potential issue with the Nuplazid. You know, typically I would tell people sometimes it's worth trying. And if it doesn't work, it doesn't work. We move on, we try something else. But I think in some cases, the potential side effects are just potential until you know and give it a try. And usually if there's a problem, we stop the medication and move on. So I just kind of keep that in mind. There's a question here, when a clinical trial and are part of a control group or have placebo, how are your current medications adjusted? So this is a really, really important question when it comes to Parkinson's disease. So there are some studies that will dictate to the investigators what they can and cannot do with the Parkinson's medications. There are other studies that say, you do what you need to do with the medication. We just want the patients to feel good. And so it really depends on the study. Arguably, if you are in a study and they are monitoring how much Parkinson's medications you're on, you shouldn't change it without talking to the investigator so they can keep track of it. But you really need to be clear up front when you're thinking about a study, whether or not they want to adjust your Parkinson's medications or if they have rules against it. There are some other studies that you come in without Parkinson's medications at all, and they look to see how long it may take for you to then start something like Sinemet, for example. And so to keep in mind that that's another option that's out there for a lot of these studies. I would say in general, most studies don't stop the Parkinson's medications, but they may have some recommendations as to how things are adjusted or not. So keep that in mind. Okay, so there's another question about current treatments or drugs that are exciting. So, I mean, I'm biased with the gene therapy, but I think that's really exciting just because it's a completely different way of trying to address these diseases. There are some other immune modulating drugs out there. There's like a vaccine for alpha-synuclein that's being looked at. It's done fairly well in early studies. So for multiple system atrophy and Parkinson's disease, they're looking at this. I think that's interesting. We'll see where it goes. A lot of them are just now getting out of phase 1 and looking at phase 2. And so again, that's that big leap for Parkinson's studies where they really got to get past that phase 2. So we'll have to see how that looks. There are some new, I'm thinking specifically of like constipation. There are some interesting drugs out there for that. I really would like to see the field getting away from new ways of delivering levodopa. I know there's a need for it out there, and that's a niche thing, but it would really be great if we could start focusing on a lot of, again, the more of the non-motor symptoms. And I see that happening, but we're just now seeing that uptick. Again, part of the problem is that we don't have good models of the non-motor symptoms in the animals. So it's really hard to then get that through the early lab side of things and into people when we're not even looking for it in the lab per se. because the Parkinson's models we have don't do a good job of looking at the non-motor stuff. There's a question about adding entacapone or using extended release formulations, I'm guessing of Sinemet, to elderly patients. So it gets really tough to know what's best. It really depends on the person. The extended release Sinemet is very difficult in that it is very erratic in how it's actually absorbed by the body. And so generally, we don't like to use the extended release formulation during the day when you really need to know when that medication is going to kick in which case, I think, most movement specialists would move on to entacapone to really, again, the idea here is you're extending the duration of benefit of Sinemet. This is the whole point of things like Rytary and some of these other added medications. Again, you're just trying to get more benefit out of one dose of Sinemet. So, to be careful though, depending on if you do extended release versus entacapone, you think about this in elderly, but it depends on the person, each individual, and in their particular situation. Yeah, okay. So I just wanted to point out, so Sherry Mazovsky is the main head for clinical trials at UPMC. I see her email there. She's such a lovely person. Like, just reach out to her. If there isn't anything that works for you right now, there is a registry, so you can even just put yourself on her radar. So that if something comes up that looks like you might be a better fit for, she can then contact you down the road too. So to keep that in mind. UPMC is bringing in new studies all the time. So Sherry's really good. She's awesome. So she's a really good contact to keep in mind. Yeah, so there's this Piccopone. So this is another add-on medication. Again, there's a lot of methods that are being tried to really extend the benefit of levodopa. And so I still really want to see us, doing better than just, improving on levodopa, but there's still a need out there now for different ways of delivering these medications. And another example I'm thinking of right now is like the inhaled levodopa. So for these like sudden off periods, so that's another new type of formulation. But again, just from where I'm sitting, and I know my husband shares this, like, it's not actually addressing the underlying problem of the disease. And this is really where we see changes in the research, really focusing more in that direction. But really these things need to be happening in parallel. So we need to keep getting better with what drugs are out there, but really at the end of the day, to make any headway in Parkinson's, like, we need to try and stop this in its tracks early on. So.
And that's something I want to add to the your comments on the question of what out there is exciting, and the stuff that's coming up. Of course, again, I'm also biased towards gene therapy, working in a gene therapy lab, and I've seen the data. It's really exciting stuff. And yeah, the antibodies to alpha-synuclein are doing well and moving into phase 2. But the reality is, when we look at the pathology of PD, each of these new things that's coming out, yeah, now we're starting to get into altering disease course, but we're still largely looking at treatments that look at one thing, like encouraging dopamine regrowth, or getting rid of the alpha-synuclein that's already there, or encouraging regeneration in the gastrointestinal tract. I think what's really exciting isn't any one of these drugs. It's the potential that if we get several of them through the door, how well will they work in combination? Can they work in combination? And we might see some combination therapies coming through. We don't have anything out there yet, but it'll be really exciting once they start showing up.
Yeah, cancer had to go through this. I don't know why it's taken Parkinson's research to take so long to come to the same conclusion, but it's going that direction. Again, it's a complicated disease. It's not just the brain that's affected. It's not just that tiny little substantia nigra. There's so much more, and really why I think there's more and more attention being paid to hitting it from like multiple angles to really make some headway here. I just wanted to answer this last question from Bill. So this CoQ10. So maybe some of you remember, but CoQ10 was like, it was a hot thing for many, many years. And they finally did a bigger study, and it just didn't look to have that level of protection that we all thought was going to be there. So happens to be that there is actually some interest in reviewing and redoing this study. So again, looking at really, I mean, super high doses of CoQ10, we're talking like 4 grams of this stuff, like really high doses, but looking at a very, very specific type of Parkinson's patient to see if there's a subgroup that will be able to benefit from this more than just what they did initially was like all comers. If you had a Parkinson's diagnosis, everybody was treated the same. And maybe that's just not the best way to be studying Parkinson's disease in people. Stand by. We'll see what happens. Thank you, everybody. It's always good. It always gets me revved up talking to you guys.
Hey, yeah, thank you, Drs. Van Laar. We have come to the end of another hour. So thank you all for participating. And we'll see you next month in the diner. How do you like that? That's my best announcer voice too.
Yes.
I feel like I need like a screenshot of like the 50s diner behind me.
Yeah,